Hi, I want to talk about a post-translational modifications. PTMC. PTMC can play important roles in many biological processes, such as signal translations, immune systems functions, and cell cycle regulations. These regulations of PTMC has been implicated in a number of diseases, including cancers, diabetes, and neurological disorders. PTMC refer to modifications to proteins that occurs after the protein has been synthesized and translated from its corresponding genes. These modifications can take many forms and can affect protein functions, localization, stability, folding, and activity. There are many different types of PTMC. Phosphorylation. Phosphorylation involves attaching a phosphate group to protein residues by enzymes called kinases. Whereas dephosphorylation is done by phosphatase. The most related amino acids are serine and threonine. Binding of the phosphate groups in some proteins activities them, and some proteins are activated by removing the phosphate group.
Acetylation. This involves the addition of an acetyl group to a protein. This can be done by enzyme called acetyltransferase or modified by abundant acetyl-CoA through non-enzymatic mechanism. Convert lysine deacetylation is catalyzed by lysine deacetylase, result in transcriptional activation due to destabilization of DNA histone binding, as acetylation of lysine in histone protein neutralizes its positive charge, which prevent the formation of salt bridges with the negatively charged phosphate backbone of DNA. In the figure, acetylation tail in histone protein resulted in loosely packed DNA.
Methylation. The addition of a methyl group to a protein. This can be done by enzyme called methyltransferase. Lysine and arginine are the two main amino acid target residues in methylation. One of the most biologically important rules of methylation is histone modification, which include transferring the methyl group from S-adenosine methionine to histone protein via methyltransferase enzyme. The picture shows the role of acetylation. Regions with high transcriptional activity are loose packed, and methylation regions with the low or no transcriptional activity are densely packed.
Glycosylation. Glycosylation has many functions. بدنا نحكي بعلاقتها بالبروتين بمعنى glycosylation aids in protein folding and stability and localization and services as a quality control checkpoints for properly folded proteins. Enzyme glycosyltransferases link sugar to proteins while glycosidases remove sugar from proteins. About half of all cellular proteins are glycosylated, most such as secreted proteins and structural proteins. Ubiquitination. This involves the attachment of a small protein called ubiquitin to another protein. This process is usually associated with the degradation of proteins, and it is regulated by enzymes called ubiquitin ligases. As shown in the figure, protein-degraded E1, E2, E3 enzyme cascade was intervened and are regulated by ubiquitin ligases to form the protein. Trimming of proteins is the removal of part of the peptide chain and works to activate or inhibit the protein. Activation of a protein by converting them from zymogenes to enzyme like digestive enzyme trypsinogene trypsin and activate some hormones that are made ineffectively, such as insulin. As shown in the figure, preparoinsulin and paranosol and insulin